So let me start by thanking the organizer for having me. I've learned a great deal from the BPPB webinars. The YouTube channel is one of my favorite YouTube channels, so it's great to be part of this. So I'm going to talk about two topics of active biological system, or light production and adaptive morphodynamics. Before I start, maybe let me thank the group of people who have helped me with the results that I'm going to show today. In particular, I want to acknowledge the work of Nico Shrama, who has been involved in almost everything that I'm going to show today, and also Ray Goldstein, who was the one indeed that brought me not only about the things that in the research topic that I'm going to talk today, but generally the biological physics research. The people on top are from my group at University of Amsterdam, and the rest are the colleagues across Europe. All right, so overall, one of the research fields that in biophysics scheme that my group is interested in is basically the concept of photosynthetic organisms. There are many of them. Here is the branches of tree of life, very simplified, the ones that would do photosynthesis. It can go from algae and bacteria, all sorts of bacteria, to clearly plants that we are very much familiar with the photosynthesis. And the way I look at the biophysics of these types of organisms is through two ways. It's basically overall about life interactions, but about also life absorbing light, and also life emitting light, which is something that I'm also going to talk today. There are various research on many aspects of light and life interaction overall, with many big questions. One question that many have asked for decades is that what an organism do if it sees light, in principle. And we know it ourselves. Now that the weather is warm and nice, we move towards light if the intensity is suitable for us. But if the temperature is too high, then we prefer to avoid high intensity light. And of course, we go to the shade. This is very much known not to be limited to mammals or animals. In fact, there is a large body of work on other organisms like green algae, which something that I'm also very much interested. Here are examples of green algae. This is a single-cell Chlamydomonas, which is a model organisms, and through multicellular evolutions, you can get gonium, which has more, all the way to Volvox, which has a lot of singles, or each dots are single cells that sitting and form a sphere with a lot of flagella. So what these organisms have are these tiny hairs that are called flagella that they can swim around using that mechanism that relates to many field of research in microhydrodynamics, in fact. So here's an example that is from the work of the Malprat, Mazi, Ishikawa, and Goldstein. That this is a gonium. This one is a multicellular green algae that swims. If you look carefully, you can see the flagella. So it's basically moving around and swimming in water. And they also, because of photosynthetic, they also react to lights very nicely. So what here the experimentalists have done is that they put lights on the right, and gonium goes to the right. You switch the light to the left, gonium switch to the left. So you can keep doing it. So this is phototaxis. The organism moves towards light to be able to do more photosynthesis in principle. So this is not the type of work that I'm going to talk about today. In fact, there is a BPPB seminar by Marco Polin about the phototaxis of Chlamydomonas. So if one is interested on the review of the work, there are many people who are doing great work on this topic. I can highly recommend that seminar as a summary, and also some advancement from Marco Polin's group itself. Okay, so these are about motile organisms, so they can move towards light, but less motile organisms can also move towards light and away. So the great example of it is plants. So similar to the previous experiments here in this YouTube video, the light first comes from the left, and the plant grows towards light, and then it goes to the right, and plant grows towards to the right. So this is called phototropism, and this is basically on a scale or time scale of many hours. As you can see here, and it heavily depends on the growth of the organism, in this case, plant. There are very classic words since Charles Darwin, who was interested in this, but on more recent literature, there are many groups, including Alan Growley's and also Yasmin Meraz's groups, who study this type of interaction of light and life in the context of plants or growth in general. What we have recently found out is that we discovered there are some series of very interesting physics and biophysics, not on organism level, but on a cell level. So for example, in the case of plant, what I'm going to show here is basically the leaf of a plant called Iledia densa. So what you see in each of these boxes are cells, and each of the dots that you see are chloroplasts, the organelle that does photosynthesis to generate oxygen. What we found out, very briefly, if I want to put it, is that under dim light, these chloroplasts form this monolayer to do high efficiency photosynthesis. But if the light intensity goes very high, like now, chloroplasts start to be highly motile. And this is a photo avoidance scheme. So, and the time scale corresponding here is actually much, much smaller than the phototropism, for instance, time scale. So not only on organism level, but plants also have evolved some other light adaptation mechanism to protect themselves from intense light. So here is a quantified view of this. For instance, here is under dim light, they're just sitting around and wiggling around, and you increase the intensity of light when the sunlight, for instance, is very intense. Within a few minutes, they become motile and they react to it to protect themselves. There are many nice physics that one can do, but I'm not going to talk about plants, but I'm more than happy to talk to anyone who's interested later. So the big question here is, okay, if an organism is motile, it can avoid light or can go through like phototaxis or anti-negative phototaxis. But what about if an organism is non-motile? So this is one big question that we want to answer. And how do non-motile organisms, what have they evolved to protect themselves against high intensity light? And the hero of the talk today is this organism. It's called Pyrocystis lunula. It's a dinoflagellate. So this is a eukaryote single cell. And so what you see here in this picture is many of them. So if I could, if you see my arrow, this is one of the cells, and they're relatively big eukaryotes. So there are 15 microns in one side and about 100 microns in other side. They have cell wall. So if you're mathematicians, the leading order, these are plant cells. And they also do photosynthesis. So basically, they have chloroplast, which is this brownish color here that does photosynthesis. And the cooler thing about this particular organism, which is pretty much everything I'm going to talk in this today is about it, is that it can generate light. And when you also, we've recently found out, when you shine light to them, they have a very peculiar mechanism to avoid high intensity light. And that's a major discovery that I'm going to talk about today. So I'm going to focus first on how do they shine light. If you've been lucky, you might have in a warm spring or summer been one of these, have seen one of these phenomena at a shore. This happens all around the world now. This video is particularly from a beach in Australia, but in Caribbeans and nowadays even in Europe, you can actually see quite a bit of this phenomenon called algal bloom. But these dinoflagellates, there are trillions or billions of dinoflagellates that, because the temperature goes high, they can multiply very quickly, and they can carry all the way to the shore. And as you can see, they can generate this rather high intensity blue light. So it's a glowing algal bloom, and these are marine organisms. So this is ocean, basically, or sea. Something that was very interesting for me when I saw this video first time was that, it seems like when you put your hands or feet inside water, that's when the light production happens. This seems to be a mechanical cue involved, which I'm going to talk about it shortly today. There are many organisms who can do this, many dinoflagellates. They come in all forms and shapes. Dinoflagellates in general, some of them are photosynthetic, many of them, and some are not. Some of them are actually motile and some are not. Here are three bioluminescent dinoflagellates. Pyrocystis fusiformis, which looks like a needle. Some are more like a spherical shape. And this is our favorite dinoflagella, Pyrocystis lunula, which also generate blue light, but has this green brownish chloroplast to do photosynthesis. Okay, so it looks like this. It looks basically, it's called Pyrocystis lunula because it looks like moon. Unlike moon, it's non-motile, I told you already. So it's a great example of cells to do a lot of experiments in the lab because you grow them rather easily and you can put them on the microscopes and they don't move, so you can do whatever you want basically with them. They have a rather complicated cell wall and it's a multi-layer of cellulose and which is below it. There's this lipid bilayer with all sorts of molecular machinery that leads to various types of signaling pathway inside the cell to do multiple things. It is very much known the biochemical pathway that leads to bioluminescence in dinoflagellates. It seems to be known that upon some stress, like mechanical stress, you start some sort of calcium signaling inside the cell that leads to action potential in the vacuoles. And there's this organelle called syntelin that carry bioluminescence organelles, which is encapsulated within these vacuoles. And when the action potential happens, seems like hydrogen gates open and the pH in this organelle drops. And that makes the environment suitable for this chemical reaction, which is very, very known for biophysicists called luciferin-luciferase interactions. You basically burn the substrate luciferin with the enzyme luciferase. And what you get out of it is oxyluciferin, but more importantly, light. Okay, this is also known as cold light production. Like I said, there are various works on for decades on biochemistry of bioluminescence, led to major discoveries like the discovery of GFP. On a more biophysical side, the work of Mike Latz in Scripps in University California, San Diego, has been pioneering, and he and his colleague has done great works on the past decades. So I can also highly recommend if someone wants to start on bioluminescence research. Okay. So the question we wanted to ask was the first question was on a single cell level, can I probe how mechanics leads to light production? So this is a experimental setup that we built. So everything is on the microscope. This is the cell. To remind you, this is almost 50 to 100 micrometer size of the cell. It's trapped between two pipettes. So this is the first pipette and the second pipette. One of them is bent. And it's bent because it basically looks like an AFM tip now. So you basically, when you push the cell, you also deform this and you can have an estimation of the force that you're imposing on the cell. Each pipette is attached to a micromanipulator, so you can control how much and how fast you can deform the cell. This was very critical. And basically what we do, we squeeze the cell in between the two pipettes. And what you have to look carefully is this cell here. And I'm gonna quickly squeeze the cell between two pipettes. And when you do this, it generates light. Okay, and the idea is to repeat this in a very controlled manner. So here is the magnified view. This is one pipette. This is another pipette. This is the cell in between. This is, maybe I should say it right now, the circadian clock of the cell is highly important. This is day-night for the cell in principle. And what I'm going to show you is, again, squeezing the cell between two pipettes. Squeeze it and it generates light. And you have to look at the time scale, which is extremely fast. You're talking about tens of milliseconds that this happens. So I'm not going to speed up the movie. You can keep doing this. And if you want to look at it from physics perspective, your active solid body generates light for you upon mechanical stimulation. At some point, the cell runs out of the substrate to burn. So you basically stop generating light. Okay, so here is the same view. You have this cell, your single cells. What we've done in the experiment, we can very accurately measure how much to deform them delta and how fast to deform them delta dot. In the rheology or mechanics language, this is a strain and a strain rate in your system. And then what the observable of the system is this time-dependent light production every time that we squeeze it. So at some point, you get this signal that goes up and down. One first thing that we have found is that almost always, or not almost, always in our experiments at least, to generate light, you had to deform. And it's quite a bit of deformation that you had to pose on the cell to generate light. So not by just scratching or shear, we could not generate light production. So it seems like that the deformation of the cell wall seems to be a critical point for the cell to generate light. Okay, you can quantify these experiments very nicely. So each of these peaks that you see is every time in the video that I showed you when we poke the cell. So you poke it, the light intensity goes up. This is in arbitrary units. It goes up and then it decays down. You squeeze it again and it goes up and it goes down. And at some point you run out. If you plot it in a dynamical system form as in a phase portrait, so in a derivative of light versus time and intensity, you get these loops. So basically, every time I poke the cell, it generates light and it goes back. And at some point, it goes to this attractor 0, 0, which is when you run out of fuel. And this is out of repetition from first poke to the last one. It was important to get these loops because what we found out that the shape of these loops are self-similar. So independent of how you deform them or how fast and how much you deform them, you always get a similar shape. So if I scale this with the maximum values, you get one loop. I'm not going to talk about theory, but that led us to actually form a very simple linear theory, which if you want, I can talk about it after the talk, that to get and quantify bioluminescence of the system. What was more important maybe was this physiologically important fact that we found out. So if I look at the maximum intensity as one parameter to observe and plot it as a function of how much I deform the cell, strain delta, or how fast I deform the cell, delta dot, this is the map that you get. Each of these columns are average of many repetitions. So these are really a couple of weeks of experiments because we wanted to do it in a proper circadian clock time, which is midnight for them. And each column is basically multiple experiments. You get the average, and it's very clear that if your deformation is small, you do not generate much light. If your deformation rate is very small, you also don't generate much light. You only get enough light production when both of these are high. And this is important because evolutionary speaking, this seems like the light production in dinoflagellates is correlated with, or is basically a defense mechanism, and seems like the cell response to mechanical forces, either a small fish coming to eat them or anything else, is such that they only use this very valuable substrate if the danger is actually real, if something deformed them very quickly and also so much. And that you can see it in this diagram. So this was again, I can go to in the question and answer much deeper to what we found about single cell bioluminescence. More recently, we are now going to the continuum level when we have millions of bioluminescence cells, and we try to understand if there is any collective light production, etc. Okay, but I told you already that this cell not only produces light, but also responds very interestingly. Like, you shouldn't forget, this is non-motile, so they go everywhere that the water brings them. I mean, okay, maybe I should note that they can regulate the buoyancy and go in the vertical columns, but in anything that I show, there is no such phenomenon. So something that we more recently we try to understand is that if they are non-motile and the light intensity goes very high, which if you do a little bit of survey on where is the natural habitat, you can see that the light intensity actually fluctuate quite a lot. So the question is, how do they respond to this fluctuation? So these experiments I'm going to show you much better quality of videos later, is pretty much the first experiments we've done. And this is chloroplast during the day. Sorry, this is dinoflagellates during the day. So at days, there's no point of doing bioluminescence, so they do not do bioluminescence. They actually do not carry luciferin to do bioluminescence during the day. What they do is that they have chloroplast, which is this grayish, liquidish material that is all over the cell. And they're basically sitting there and they try to do photosynthesis. And imagine now, clouds go away from in front of the sun, and the sun has a very high intensity. And what happens is that what we observe is that they shrink their chloroplast. Okay, so during this is the normal situation they have to be. Within only very few minutes, the whole chloroplast shrink to the center of the. And when we saw this video, it raised so many questions. I mean, first of all, how do they do this? And what's the mechanism behind this? And the second question, which took us a while to answer, was how do they actually do it? So if I look carefully at this video again, in the beginning, chlorophyll is everywhere inside the cell. So if it's this 100% area, and if I go quickly to the back, so the volume reduction is almost 50%. So the main question we had is, where does the liquid actually go? And if this puzzled us for a very long time. So first thing we did, we repeated these experiments in a lot more careful and controlled way. So this is the cell sitting, and what I'm going to show you is that we shine light to the cell and look at where the chloroplast goes. So this is, again, this gray ish medium that you see is the chloroplast inside the cell. You shine light, now it shrinks. You remove the light, it goes back. You shine light, it goes back. So it really looks like an active spring. So when you put the you shine light on it, it shrinks, it contracts, and then you remove the light, it slowly comes back. And you can keep doing this and play with this harmonic oscillator if you wish, just actively control it externally. So you can repeat many of these experiments and extract information like this. So what I'm showing here is the area of this of the chloroplast divided by its maximum or initial area. So that's why it's 1 at T0. And it stays at around 1 unless you shine intense light, and it goes down, and you remove the light and it relaxes back with a different time scale. You can keep doing this, and if you let it relax for much longer time, it relaxes back to where it was before, in principle. But again, the time scales are rather important in the system. We try to model it on a 0 dimensions, and having background in mechanics and rheology, something that this looked like to me was some sort of active solid in a sense. So you can given these time scales, the various time scale of relaxation and stimulations, it looked like a viscoelastic solid to me. So we thought, okay, if this is viscoelastic, now all we need to do is to add an active element. So the very known zero-dimensional model in viscoelastic solid is a Kelvin void. So here is an active Kelvin void, if you wish. So this is a spring in parallel to a dashboard, but now there's this active element term which you could put there or here. It doesn't matter really. The nice thing about this model is it's extremely simple. So you can write it in linear form. There is now tau 1, which is the viscoelastic time scale of your system. And the question is, what is this FT? So in our biological system, we know FT comes with some sort of chemical signaling. For instance, calcium most likely is important here, which you can show by this C. So you have to solve another linear differential equation for that. The major question is how C and F are related to each other. And this is something that, although despite being very simple, we learned from the model. Because initially we thought with just one signaling equation, we can actually, or concentration equation, we can fit through our experimental data. We found out we actually need 2 additional time scales in the system, which I'm showing by tau 2 and tau 3. Later on, when we look at the genetic information from the dinoflagellates, we found out they have 2 photosensors with 2 different time scales. And that seems to be, in fact, corresponding to the time scales that we get when we fit through the experimental data. So here's what I've shown you before. And this dashed line are basically the fit through the experimental data. What you can learn is the time scale of these models, and again, how you can get some information about overall time scales involved in the process of contraction of chloroplasts and coming back. Okay. However, I told you this, but something I haven't told you, and it took us a while to understand, was how all these chloroplasts actually contracts to the center. I mean, if it's an incompressible liquid, you cannot contract them. It's surely not compressible liquid. That's not a thing in those scale and biological system. And if it was a normal solid also, if you contract it, the Poisson ratio is positive, then you have to get contraction on the other side. But there is cell wall. So this question of how under confinement of the cell wall you can contract this material by 40–50% was a major question to answer. So the way we answered that question was through confocal microscopy. So this is basically what I'm showing. Maybe I pause and I explain what is T0. So this is how the cell looks like during the day. And what you are seeing is the autofluorescence of the chloroplast. So this is 3D image of the cell. Done by Nico very beautifully. And what we observe immediately is that it's not really a homogeneous structure. It's really a holy structure, meaning that you have this reticulated system. It's a network of chlorophyll attached to each other and forming this very complex network that if you shine light to them, then it contracts. And as it contracts, it basically buckles and closes these holes. And that's basically was the secret of the dinoflagellates, to be able to shrink all these liquids to the center. The only way to see it was to actually look at the whole 3D structure of this reticulum. Okay, this raised many, many questions that I briefly cover a few of them. So topologically speaking, if you like topology, this is basically a topological surface that is active everywhere and is contracting itself. And you can look at it from many ways. If, for instance, if you want to characterize it, almost all the cells that we scan have a genus of 100, meaning that it has 100 holes in it that is squeezing it down. It goes down, so you close the holes. I will show you in a second quantified values as you contract. But when it comes back, you maintain the same topology. There are also geometrical arguments that you can make, the way that how these holes close. If you look carefully on some of them, you can see that they basically buckle this network of chloroplast to close the holes. And this buckling, this inward folding, is basically something that is important for such a mechanism to observe. Okay, you can quantify this, and the way you can quantify it, this is a video Nico has made. And maybe before going there, I mentioned that you might have many questions about what is the underlying biochemical mechanism, and there's some work in the literature talking about the importance of the actin filaments and microtubules, but seems to be mainly actin filaments that are in charge of the this type of photoavoidance. Okay, so this is how we analyze such a network. So this is, again, one example. What Nico has done is it fits a network through this, and you can see where the dots of each network are. And now you can look at the dynamics of network as it evolves. So maybe I quickly play this video again. So this is directly from the confocal microscopy images. You fit a network through it, you see where the nodes of your networks are, and follow the node positions over time. So this is how we analyze geometry and topology and the rest of the features of this network. So here are some important results. What I'm showing here is the mean square displacement of each of nodes of this network that I just showed you. And this is basically delay time. So it is over time. And what you can observe is that most of the nodes of the network are basically diffusive, except a very few. And if you look and correlate it with the space, these few nodes are at the very tip of the cell. So what the cell does is basically, and they're in principle ballistic, so ballistically move the tips of the chloroplast networks to the center, and that is enough to be very fast response as a photo avoidance over time. You can do now a lot with this big data that you can collect. So here is the blue light stimulations. The cell contracts all the way, the chloroplast contracts inside the cell to the center, and it does go back if you just let time. You should note the time scale, that this is within few minutes, and this is almost much larger, so it's like an hour or so. So the relaxation of the chloroplast it takes much coming back, expansion takes much longer. So here's a distribution of on the bottom left edge length over time. So what you see is that, again, you have the important part is this tails of having a long length, which are corresponding to such strains here, that shrinks very quickly. And that's all you need to actually reduce the chloroplast area when the light intensity goes up, despite that the average remains the same, because most of these structures in the center, and you can also correlate it with the orientation over time. But maybe more importantly, I focus here on the on the topology of the system. So like I said before, the number of holes in these networks for almost all cells remains order 100 or around 100 before and after a stimulation. So you have this cell or chloroplast network structure that has many holes, hundreds of them. They shrink and collapse, but when you let it come back, the shape of the cells, if you compare this one on the right and the left one, is different. So the details are different, but the number of holes seems to be always pretty much the same, which raise many questions that, is there some sort of optimal form of topological surface or not, which is hard to answer. Okay, last thing I want to show about this light response is that we were very much curious to see how local or global the interaction, such a stimulation and response to light is. So here I'm going to show a very similar experiment. So this is the chloroplast network. And instead of shining light everywhere to the cell, we're going to do something that is physiologically really not relevant. So basically, this is a lab experiment in a way that we're going to poke just a corner of the cell and see here in this box, very small amount, and see how a cell react to it. And this is what we've observed. So if you break the symmetry in the way that you stimulate the cell response also is symmetry broken. So in the sense that it seems like that the sensing is highly local. So it probably the concentration of whatever chemicals that the cell produce, most likely something like calcium, seems to be higher here. Therefore, there's more activity on the side that you stimulate the cell. Unless you see some little bit of activity on the left side too, which is pretty curious. So if you do some back of envelope calculation, seems like calcium diffusion is enough to get something like this, but how cell regulates the concentration of calcium inside something or other chemicals is not very clear to us. Now you can shine light to the center, and this is something which looks like a puzzle, because everything I showed you so far, whenever we shine light, the chloroplast avoid that because the light was too much. Now I'm shining light to the center of the cell, and chloroplast actually moves towards it. And you might ask why it happens. So why chloroplast moves towards the cell? And we think the reason is that, something that I didn't tell you, the nucleus of the cell, so eukaryote cell, is located at the center of the cell. So this is most likely some sort of action of the chloroplast to protect the genetic information inside the nucleus. Okay, so with this, let me go and quickly conclude and in a very high level. So what I tried to show you today was that in general, it seems like the evolution led to many very interesting photo adaptation mechanism, and photo avoidance is what I talk about, but also on the other side, using light to do something like in bioluminescence. And I think using the new biological physics tool, we are just about to understand some math and physics related to this biological phenomena. And there's a lot of more questions, and probably with what I've shown today, I raise more questions than answering any question. And I think this is a great opportunity to go to. These are some of the references. I didn't really talk about plant chloroplast, but there's a recent publication from my group. The work on single-cell bioluminescence was done during my postdoc at Ray Goldsmith's group in Cambridge. And the flagellate chloroplast we recently submitted. So any feedback from this community is highly appreciated. And I want to end the talk with two advertisements, if I may. First thing is that for the next March meeting, which is going to happen in California with Orit Pelek from University of Colorado, we are organizing a focus session for the first time on light and life. So I highly encourage people who work on the intersection of optics, life, and light, and also any biological application, to submit their contribution there. We would be very happy to see this focus session grow over time. And last thing, that I have a postdoc position in my group related to this topic or anything else. So I would highly appreciate if good candidates for such topics. And with that, I would be very happy to take any questions.